All right. Two more overview reactions to do and then we're done. I think it's been confusing for me to try to talk you through something that's not in front of you. So we're just going to go straight to the complicated example from your mechanism handout and we'll work from there. By the way, when I teach this to organic chemistry students, what we have done in the past 3 lectures probably takes me 3 weeks to do. Now, we do a lot of examples of stuff that you do in the lab, so there's a lot more stuff that we're not doing here, but this is a lot to try to absorb. So it's okay to feel overwhelmed, and frankly, you should feel kind of awesome if you're if you're following along and if it's going okay. All right. So what I'm going to show you now is almost exactly like the aldol reaction. It's called the Claisen reaction, and the only difference between the Claisen reaction and the aldol reaction is in the identity of the electrophile. The nucleophile in an aldol reaction is still an enolate, negatively charged on the alpha carbon. You got lone pair electrons on the alpha carbon. We're showing this reaction after we generated the enolate. I don't know why I chose to do that. If you needed to push arrows to show the formation of the enolate, you would start with its conjugate acid, the carbonyl compound. You would use a base to remove the proton from the alpha carbon. But establishing that as the enolate, the other molecule is a thioester, which is what level of electrophile? Ester is 1, thioester is with the sulfur, and that's, ester is 2, oxygen ester is 2, sulfur ester or thioester is 1. So that's one that you might be confused on. It's the thioester with the sulfur in it that's level 1. You've got in the thioester carbonyl carbon, carbonyl oxygen, and then thiolate leaving group. Okay? So in this reaction, what will happen is nucleophile attacks carbonyl carbon in the usually expected way. This shouldn't be new or surprising at this point. And you break the pi bond. In the intermediate, here is where the nucleophile ends up. What used to be your enolate is right here. The electrons that were on the negatively charged alpha carbon end up in this bond here in red between the purple alpha carbon and what used to be the blue carbonyl carbon. Here's the carbonyl oxygen that has the negative charge. Here's the thiolate leaving group. You can tell that that's a good leaving group because you can imagine what it would look like when it left. This would be the coenzyme A group with a negatively charged sulfur on the end. Its conjugate acid is a thiol with a pKa of 8. And in general, if the conjugate acid has a pKa of 16 or less, we're going to say it's a decent enough leaving group. Electrons on the negatively charged oxygen kick down, thiolate leaving group leaves, and there is your product. Your product, this portion of the product used to be your nucleophile. This portion of the product used to be your electrophile. The coenzyme A leaving group is gone, and in red is the new bond that you made. So this is a Claisen reaction. Yeah.
We're supposed to assume that the 2 hydrogens are being taken away, right?
The 2 hydrogens in the enolate? No, they're still there. They're implied but not shown on that alpha carbon.
Oh, is it? Okay.
Yep.
Are we missing a hydrogen on our paper in the negative charge?
Here?
I don't know if we were about to, I don't have the first hydrogen in the negative charge, so I just don't know.
You don't have this one or the negative charge?
Yes.
It's on the mechanism, not the quiz.
Oh, shoot. Okay, well, let's look at the quiz then.
So I'm just curious where you would pull the electrons from.
Okay. Yeah, so what, this is the I got too many things in different documents, so my apologies. I'm embarrassed now. Okay, this is what you're, Cambry, is this what you're looking at?
Yes.
Okay. Yeah, so in the quiz, what I had you do was not start with the enolate, but instead generate it to begin with. And then we attack the carbonyl carbon. So yeah, my apologies. If you're looking at the quiz, you've got the same thioester that's level 1. You've got the same leaving group. You have a molecule here, which is going to be an enolate. Here's the negatively charged oxygen in the tetrahedral intermediate. Here in red is the new bond that we made. And then here in green is the good leaving group. Then electrons kick down and leaving group leaves. And there you go. And if, by the way, you don't like having that thiolate there, if you want to, you can protonate it with the conjugate acid of your base.
Are we recording?
Yes. Surprisingly, how did that happen? I have no memory of pressing that button. But all right. Okay, other questions? Yeah.
Wait, what was the leaving group in the middle?
The leaving group is this thiolate. It leaves as this group. Yep. And you can tell it's a good leaving group because its conjugate acid is a thiol. And if you look up the pKa of a thiol, that's 8. So that's less than 16. That's sort of where our dividing line between okay leaving groups and not okay leaving groups. All right. We'll see that reaction. That reaction is involved in the synthesis of fatty acids. So if you have an excess of acetyl-CoA, your body does this reaction and starts to make longer chains that eventually get turned into fatty acids. And then the last one on this list is a decarboxylation reaction. So before we do this on your quiz, open up a sheet of notebook paper and I want you to draw a simpler example. So this last reaction is really important in the citric acid cycle. It's called decarboxylation of a beta keto acid. And this is how the CO2, this is one of the ways how the CO2 you exhale is generated. So I'm going to draw a carboxylic acid. And on the beta carbon, so carbonyl carbon, alpha carbon, and beta carbon, I'm going to draw a carbonyl group. This is a beta keto acid. It's a carboxylate. And actually, to be consistent, I've called it a beta keto acid. If you want to be technically precise, we could call it a beta keto carboxylate. Now what's going to happen here is somewhat bizarre, though it follows principles that we've learned so far. We're going to think about the carboxylate. Normally we haven't done chemistry like this at a carboxylate, but the presence of a beta keto group changes this carboxylate. So I want you to relax your eyes a little bit and see whether you think this carboxylate imitates kind of a tetrahedral intermediate. In other words, in a tetrahedral intermediate with a leaving group, you've seen a negatively charged oxygen with electrons on it attached to a carbonyl carbon, where that carbonyl, what used to be the carbonyl carbon, is attached to a leaving group. And you've seen an elimination step where Electrons on that negatively charged oxygen kick down and the leaving group leaves. And that pink oxygen is what I'm going to call the kicker offer. And when your kicker offer is adjacent to a leaving group, then electrons can kick down and the leaving group can leave. My contention today is that this negatively charged oxygen from a carboxylate can be a kicker offer. The bond to be broken is this one between the alpha carbon and the carbonyl carbon, and that would make this over here a leaving group. Now, how is it possible that that's a leaving group? Let's just try and see what the leaving group would be, and then see whether that's reasonable. When a kicker offer kicks off a leaving group, electrons kick down and the leaving group leaves. Notice what becomes of the oxygen of the kicker offer. It's now one of the oxygens of carbon dioxide. CO2 is a gas, and gases rapidly bubble off away from the leaving group. And so this reaction does not go backwards, it is irreversible. Let's draw the leaving group. We've pointed the arrow to the alpha carbon, meaning the electrons from that red bond between alpha carbon and carbonyl carbon must be a lone pair on that alpha carbon. That intermediate is an enolate. In other words, the leaving group is an enolate. Now, enolates are okay as leaving groups because they are resonance stabilized. And we've drawn the resonance structure for an enolate before. It looks like this, where the negatively charged, where the negative charge is out on the oxygen rather than the alpha carbon. Okay? After that, the enolate can readily be protonated by some acid or the conjugate acid of some base. We'll just make it some acid. Proton transfer reaction, and what you're left with is the 3 carbons from your leaving group minus CO2. That's called a decarboxylation reaction. Go ahead.
So why, when you when you give the carbon, not protonate, when it's it has 5 bonds, why isn't it a pi bond that would break? Is it because
What has 5 bonds? I'm sorry.
So when you bring the electronegative oxygen's electrons far left onto the carbon, yeah, you make it up a bond, and then the carbon has 5 bonds. Why wouldn't when its pi bonds break if it's
So you're saying that when you do this, you need to break something, and your question is, why don't you break this one?
Yeah, well, I know why you wouldn't, because
If you do that, that's just the other resonance structure for a carboxylate. So what I think you're arguing is that doesn't the kicker offer oxygen already do this? Yes, that's just resonance.
I guess what I'm saying is the reason it wants to break the carbon-carbon bond because the gas is a stabler form?
Yeah, so this is a great question. What makes this reaction go forward so profoundly? There's going to be no math here, but we're just going to talk qualitatively. I think I've said this before. Whether a reaction is favorable refers to 2 things. First, enthalpy, which is, are the bonds formed stronger or weaker than the bonds broken? And then delta S, which is disorder. 2 things can drive a reaction forward. The bonds you form are much stronger than the bonds you broke, or disorder is much larger. Now, when you take a molecule that is 1 and split it into 2, disorder increases. When one of those is a gas, disorder increases a lot because gases are way less ordered than liquids are. And so entropy drives this one forward. Yep. Other questions about decarboxylation? Yeah.
So since this is entropy driven and everything, would it be
Yes, this is spontaneous. And the best way that you can determine when it's going to happen is you look for a carboxylate that has a beta keto group. It's only the presence of this beta keto group that allows your leaving group to be an enolate. Just for fun, let's draw a carboxylic acid that lacks the beta keto group, okay? And let's just ask ourselves, what would happen if the pink negatively charged kicker offer attempted to kick off the negatively charged leaving group on the alpha carbon? What would that leaving group be? It would be this molecule. That is the conjugate base of an alkane whose pKa is 50. In contrast, the enolate's conjugate acid has a pKa of 20. So it's much, 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 much better for the enolate to be a leaving group than it would be for just this anion to be a leaving group. So what I just showed you down here does not happen because this is such a cruddy leaving group. You absolutely need the ketone on the beta carbon so that your leaving group can be an enolate. The pattern you're looking for to let you know when decarboxylation can occur is, again, the beta keto carboxylate. All right? So with that, let's turn to your quiz questions. And let's do that decarboxylation of oxalosuccinate. This happens in the Krebs cycle or the citric acid cycle in your mitochondria. Some of the CO2 you're breathing out now while hyperventilating because you're sick of this class comes from this kind of thing. So just look at the starting material and see if you can identify the beta keto acid. It's going to be tricky because there are 3 carboxylates in this molecule. Only one of them is beta. Only one of them has a beta keto group. Okay, did you find the ketone? Where's the ketone?
Small center.
Right here? Okay. That's the ketone. So I've got carboxylates 1, oops, that's not a pen. 1, 2, and 3. Which one has a beta keto group? 3. To determine that, give C is the carbonyl carbon, A alpha is the alpha carbon, and beta is the beta carbon. All right? Good. So there's the ketone. Now, in a beta keto acid, where is the kicker offer? In the beta keto carboxylate, which atom is the kicker offer? Yeah, it's the negatively charged oxygen on the carboxylate. This is a little bit tricky to find. You remember to start with the carboxylate, then here is the carbon that's going to be CO2. This is the bond that's going to be broken, and everything on the other side of that bond is the leaving group. Notice that if the leaving group left, the lone pair electrons would be on the alpha carbon where they'd be stabilized by resonance. Okay, questions about how we identified those pieces? Yeah.
How do you identify the beta keto?
Okay. Anybody want to take a crack at that? How do you identify the beta keto carboxylate? Where'd you start? Go ahead.
Well, I saw 3 carboxylates.
Okay.
And then I said, like, which one is 2 lines away? Like, there
Okay.
should be 2 lines in each of them, obviously.
Okay, so you first identified the 3 carboxylates, then the keto refers to a ketone, so you're looking for a carbonyl group not involved in a carboxylate. There's only one of those. And then the CO2 that's going to come off is the one that's, you're right, 2 lines from the ketone or 2 bonds for the ketone. So you could have started here and said, okay, this is a carbonyl carbon, alpha, beta, gamma. No, too far away. This is a carbonyl carbon, alpha, too close. This is a carbonyl carbon, alpha, beta, just right. Okay, good. Others? So what arrow am I going to use then to show the decarboxylation? Where does my arrow start? Let's start with those lone pairs. They move here. That signifies the making of a new carbon-oxygen pi bond. That's the pi bond between this carbon and, let's say, this oxygen and CO2. And then I have to move the electrons out onto the alpha carbon. Or, alternatively, if I want to generate the other resonance structure of the enolate, I can do this. Okay? Turns out that's what we did to get this structure. Here is what used to be the alpha carbon when the CO2 was present. Here's what used to be the beta carbon. If I look at the product, though, the last step is a proton transfer step where electrons on the carbonyl oxygen kick down and the alpha carbon picks up a proton from some acid so we can get to the neutral product. Okay, and that's, we will see that in the citric acid cycle, the first few steps are all about getting this ketone in the right place so you can do the decarboxylation reaction. All right, I think that's it for our new reactions. Other questions? Yeah.
This is all just like a shorthand, right? Like, we're not showing all the arrows of the bond between then going the lone pairs on oxygen and that going like that on.
So alternatively, if you, what I've shown here is perhaps a resonance structure of an alternative thing you might have done. So let me just draw that. Let's see, this would have been alpha and beta. So yeah, you could draw an intermediate that would look like this. And then this would pick up the proton there. These two are resonance structures of each other. It's kind of nuts that after like 2 weeks of just like, welcome to OChem, Homo Lumo, and this is carbon, you guys are doing this. Massive, massive amount of work. All right, other questions? Sorry, yeah, go ahead.
I understand that you start the lone pairs of oxygen, but can you explain that second arrow? Like, why don't you break the pi bond between the other oxygen?
Okay, yeah, so what is it about this second arrow? You understand this first arrow. I think what you're asking is, oops, that was not the right eraser, there we go. What you're asking is, why don't we do this? And I will tell you that happens with carboxylates all the time anyway. So here is a carboxylate and it is in resonance with with this. That's actually how carboxylates share electron density between the two oxygens, and that's one reason why they're pretty stable conjugate bases. So it's not that that doesn't happen, because that's happening all the time anyway. What drives this process to happen, though, is if you do kick off the enolate leaving group in green, what you make is CO2, pretty stable bonds, and it's a gas. Entropy increases, and the reaction goes forward. So there's a driving force for this to happen, and it's that CO2 is easily removed from the reaction. Yeah. Good. Others? Okay, well, time for the sweet world of carbohydrates, sugars. In chemistry, it's really kind of lame. People that do carbohydrate chemistry, or rather, you can tell the people that don't do a lot of carbohydrate chemistry because the first chance they get to write a paper about it, they put the word sweet in the title and they think it's cute. And it was cute the first time or 100 times, but anyway, yeah.
So if there was a test question like this, would you have put the acid with like, I guess a negative charge? In the very final product, would you have put A with a negative charge?
Oh, if this were an exam, so sometimes if you use an acid, I will forget to do that, and it's.
But if it were a test question, would you make sure it would be there, or should we just be able to tell?
So you're uncomfortable with the fact that I forgot to put A minus
Just
there.
a little bit.
Yeah. Okay. Fair enough.
Because it's like ending starting point.
I'll take that under advisement. Yes, that's a fair criticism. Sorry. Good. What else? Carbohydrates. All right. So there's a couple of problems on your problem set. I think there's one of them. Let's look at that problem set and let's see what we're going to be able to get done in the time we're together versus what you don't have to do. I think you can do 9 based on what we will do today. We're not going to get to 10 or 11. If you did them already, congratulations. I don't know. Who's the one that? Yeah. There may be somebody out there. So we likely will not get to 10 or 11. So you don't have to do 10 or 11. Maybe use your extra time to ponder on the spiritual reflection question or something like that. But no, we won't do questions 10 or 11 for this problem set. I'm just going to copy and paste them to the next one. All right. In terms of 9, we'll see what we get to. You can do you can do what you can. So carbohydrates. There are 3 carbohydrates whose structures I want you to know and have memorized. And the carbohydrates that we're going to talk about have both open chain and what are called cyclic hemiacetal forms. Those are words, the word hemiacetal maybe doesn't make a lot of sense right now. That's okay. I'm going to start with the cyclic hemiacetal form, which is really easy to draw because you just draw the 6-membered ring in its chair conformation. So you do the sort of chair bow tie, and in the upper right-hand corner you put oxygen. And what I'm going to draw for you is the technical name for this molecule that I'm going to draw is beta-D glucopyranose, but most of the time we're just going to call it D-glucose, and because biology only uses the D enantiomer of glucose, we're just going to call it glucose, okay? Technically, D-glucose refers to the open chain form, Technically, D-glucopyranose refers to the cyclic form. We will use D-glucose and glucopyranose sort of interchangeably. I'll tell you, though, what the beta and the D mean as we go along. Here's an OH group that's equatorial up. Here's one that's equatorial down. Here's one that's equatorial up. Another one that's equatorial down. And then lastly, not an OH group, but what we will call a hydroxymethyl group. It's a CH2OH. I'm going to just leave those hydrogens off to avoid complicating things. If we number the atoms of glucose, it's 1, 2, 3, 4, 5, and 6. Okay. You could also draw this cyclic hemiacetal form of glucose from above, sort of a top-down view. And if you did that, it would look like this. And you should be able to interchange between the chair view and the overhead view without too much trouble. This is 1, 2, 3, 4, 5, and 6. Okay, those are the same thing. In solution, glucose exists in equilibrium with another form. This has almost exactly the same structure, only the OH group on carbon 1 is down instead of up. This is, we'll number 1, 2, 3, 4, 5, and 6. Looks like I'm running out of space, so I needed to have made my structures a little bit smaller. And then if you wanted to do the overhead view of the molecule on the right, you would draw it with that OH group on carbon 1 down, and then every other stereocenter in the molecule would be the same. One, 2, 3, 4, 5, and 6. All right. So both of these molecules to the left and the right qualify as D-glucose. The one on the left we call beta D-glucopyranose, and the one on the right we call alpha D-glucopyranose. My first question for you is, as you look at those two structures, pretend like it's exam 1. What is the stereochemical relationship between alpha and beta D-glucopyranoses? Good, great. How did you know? One and only one stereocenter is different, right? I'm going to highlight it in blue. That stereocenter at carbon 1 is different. Okay? So these two are, in fact, diastereomers of each other. Good. It turns out that because they are diastereomers that differ at one and only one stereocenter, there's another term that we will use for these. There's two other terms we will use for these diastereomers. Epimers, that's just a term. It's not really important. I'm not going to test you on that vocabulary, but I will use it in class. An epimer is a diastereomer that is different at only one stereocenter. And for these two glucose diastereomers, we'll have another term that means the same thing. It's called anomers. Anomers are epimers or diastereomers that differ at a specific carbon, and that carbon is called the hemiacetal carbon. So we need to talk about what that means. The blue carbon that I have highlighted here is the hemiacetal carbon. Here's how you recognize a hemiacetal carbon. The blue carbon is going to be bonded to an OH group, and that OH group used to be a carbonyl oxygen. And then the blue carbon will also be bonded to an OR group, and that OR group will have used to have been an alcohol. So a hemiacetal really is an addition product from attack of a nucleophile on a carbonyl carbon. That carbonyl carbon was attached to a hydrogen and a carbon. And let me show you what it looked like before the alcohol attacked the carbonyl carbon. So what we're going to do is redraw glucose in almost exactly the same way, but the purple oxygen on carbon 5 is going to be an OH group. Everything else is going to be the same, except for on carbon 1, we're going to have an aldehyde. And this is the open chain form of glucose. Okay, questions about that? Yeah.
Hydrogen, I can't remember, it's like a sort of axial, is that what it is?
Which hydrogen? This one?
The gray one, yeah.
This one is axial.
Axial down.
Uh-huh. Good. Yeah, this gray hydrogen's axial down. All right. And it turns out that similarly, the alpha anomer also has a hemiacetal carbon in blue. What's pink used to be the carbonyl oxygen. What's blue used to be the carbonyl carbon. And then that carbonyl carbon's bonded to another carbon and a hydrogen. And it's in equilibrium with the same open chain form. I'm going to draw it, and you'll see that the only difference between these two is that they are conformations of the same molecule. That is, if I rotate around the bond between carbon 1 and carbon 2, I can swing the aldehyde oxygen either sort of pointing downward or angled upward. And depending on how I do that and then close up the ring, that will give me either my alpha or my beta anomers. In other words, these two are the same thing, and they are interchangeable simply by rotation about the sigma bond between carbons 1 and 2. Now let's talk so briefly in 30 seconds about how you might show the formation of a hemiacetal from this linear form. If I want this OH to attack this carbonyl carbon, what do you think I have to do first? Excellent, proton transfer. Let's get rid of that hydrogen. We would need some base to do that. We're not going to care what. Then the electrons on the negatively charged oxygen can attack the carbonyl carbon, break the carbon-oxygen pi bond, and then, lastly, proton transfer again to get the neutral pink OH group. You can draw it this way, you could draw it stepwise. I've even drawn it, I think at one point in your notes, with a single water molecule mediating all of that. All of those options are okay. But this is how you close an open chain form to a closed chain form. I think that gives you most of the tools you need to deal with on question 9 of your problem set. The one portion probably is 9D where it talks about aldose and ketose. Just don't worry about that one until next time. We'll talk about that at the beginning of class. Okay, you made it to the weekend. You were free from me for 2 whole days, except for your nightmares. So good luck.